For our Chapter 5 mini lecture, I decided to focus on 5.1, which is all about the developing brain from a biological standpoint or point of view. So the brain development involves not just steady adding of cells, but also loss of neurons and synapses. An experience determines which cells and connections will be lost and which will be retained. We'll talk about experience later, but essentially there are 6 stages of neural development, and for this section, the vocabulary, we have a zygote, which is a fertilized egg, so when sperm and egg meet, it's fertilized, and then an embryo is an early developing individual, typically a sphere of dividing cells. Then there is the inner cell mass, which is a clump of cells inside a mammalian embryo that will form the body. A fetus is a developing individual at the stage where major organs and structures have formed, and that's about the 9th week of development or pregnancy. The neural tube is an early stage of the developing nerve system. It'll eventually form the brain and spinal cord, which I think is very interesting. Neurogenesis is the division of cells that become neurons. Cell migration is a stage of development where neurons and other cells migrate to their final position in the brain. Differentiation is the process by which individual cells become more and more different from one another and eventually get to their final destination to become the type of cell that they need to become. Synaptogenesis is the formation of synapses, and then neuronal cell death is a normal stage of development when some cells, including some neurons, just die. Synaptic rearrangement is the phenomenon of new synapses forming and old synapses retracting, especially prominent in early life. A teratogen is any substance that reaches an embryo or fetus and disrupts prenatal development, and the biggest example of this is alcohol or drugs. And then the next vocab term is fetal alcohol syndrome, or FAS, which is a condition in which children exposed to alcohol from the mother's drinking are born with distinct facial features and varying degrees of mental impairment. So the stages of neural development are neurogenesis, which is the division of cells, as said before, and then there's cell migration, when they begin to move around. Cell differentiation is when cells reach their destination and they become the type of cell they need to be. Synaptogenesis is the formation of synapses, and then neuronal cell death is neurons formed in early development dying. And then finally, synapse rearrangement is synapses dissolving while new synapses form. Because some synapses are lost, this stage has also been called synapse elimination or synaptic pruning, which actually sounds like going backwards in the process, but instead reconfiguration serves to develop more efficient neural networks. And in childhood, it's actually interesting. This is important to note that many more synapses are gained than are lost. So there's, in fact, like a net gain or total number of synapses making synapse rearrangements. And then at puberty, synapse rearrangement will produce a net loss of synapses. So once again, once sperm and egg unite, it's a zygote, then an embryo, and then a fetus, just to give an overview of stages of development. And then the next section we have experience guides brain development, which is what I said before. Experience is very important. So there's a couple vocabulary terms for this section. One being the sensitive period, which is a time during development when exposure to a stimulus has the greatest effect on a particular behavior. And then plasticity, which was actually a vocab term in Chapter 3, I believe, but it's also called neuroplasticity. And it's the ability of the brain to change in structure or function even. The biggest demonstration of how experience plays a role in guiding the brain comes from studies of formation and loss of synapses in the visual system. So unless we have enough visual experience early in life, we'll never be able to see correctly. That's something that scientists have learned. And then the studies that they've done show that visual experiences influence synapse rearrangement so that we can see. So that's why if we don't have enough experience in life, we'll never be able to see correctly. And then I included this young kittens and monkeys. So in the book, it talks about the fact that Nobel Prize winning research found that young kittens and monkeys have few synaptic connections between the eye and the brain. But as animals grow, more and more connections are formed so that by maturity, almost every neuron in the visual cortex normally gets information from both eyes. However, if the researchers close one eyelid to prevent light from entering that eye, the synaptic connections from that eye to the brain are weakened, obviously. And then if the eye is deprived of light for too long, the synaptic connections are lost forever and the animal will be blind for the rest of their life in that eye. So sensitive periods are reinforced in development in times when events can have large impacts on brain development and later behavior. So these developments, so times when events such as like visual deprivation, when that can have a large impact on brain development and behavior, that's when sensitive periods are used. And then developing brains are much more plastic than adult brains, and they recover from damage a lot easier. So the plasticity of the developing brain is seen in children's ability to recover early from damage. So when an accident or disease damages a part of the adult brain, the behavioral function is typically disrupted, and it's just different in the young, in a child's brain. So which function is lost depends on which part of the brain is damaged. As we learned previously, there are different lobes of the brain, different areas that control different things. So it depends on which type of the brain is impacted. So it's sometimes possible to recover from the lost function through intensive therapy, and that is due to rearrangement of synapses in the remaining healthy brain regions. And then, however, children with brain damage typically experience much greater recovery of brain function, and that just shows a reflection of greater plasticity. And then the most dramatic example that I included here is that when the young brain, when an entire cortical hemisphere is surgically removed to stop dangerous epileptic seizures, the plasticity of the brain in children, when this happens in children, the plasticity of the brain, when the plasticity of the brain makes the child almost always recover language and eventually develop an IQ in the normal range. So in adults, the damage to an entire hemisphere causes widespread loss of function that are never fully recovered. But it's just different for children. So younger brains in general just have a better plasticity and a better chance of developing normally after surgery or any type of damage or impact. And that's the section I chose to talk on for a little bit. I think it's very interesting because, mostly because of the experience portion when I talked about how just the more experience you have in the brain, it guides the brain development tremendously. And that's it.